Welcome to the Purr Podcast, your favorite feline-focused podcast, where we dive into all things feline, medicine, surgery, behavior, and of course, a sprinkle of veterinary gossip along the way.
I'm Dr. Jolle Kirpensteijn, feline fanatic, surgery nerd, and self-proclaimed lover of all things awkwardly cat-related.
And I'm Dr. Susan Little, proud cat vet, podcast enthusiast, and your guide to the wonderfully weird world of feline medicine. Let's get started.
This is the Purr Podcast.
I remember
This is Dr.
now.
Jolle Kirpensteijn. And
This is Dr. Susan Little.
very nice.
I
Thank you,
remembered.
Dr. Susan. So, Dr. Susan, I was reading the Scientific American, and while I was doing
The
that,
Scientific American.
I was reading the Scientific
Magazine.
American, and there was an article that described a mutation that explains the mystifying color of orange cats. And while
Yes.
I was reading that article, a name popped up that we all know very well.
Yes.
Guess what name that was?
It might have been the name of our guest.
It is. Do you want to introduce this guest?
Yes. Yes. Our guest is Dr. Leslie Lyons, who is not a stranger to the madness that we call the Purr Podcast.
Right.
Yep, I've been here before. Let me check the inside of my underwear here. Yeah, that's what it says, Dr. Leslie Lyons. That's, definitely me.
Yeah, you're the first guest who felt they had to check their underwear.
All right.
Doesn't your mommy put your name in your underwear?
Not anymore.
Some of us still need it.
Wow. So many revelations here at the podcast.
I know it's too much.
It's the mystifying orange cats.
So first I want to know, Leslie, how did you get interviewed for Scientific American?
Well, that it's mainly because they need other people that know things about cats, and of course my name comes up a lot. And then also we had done previous studies on orange, localizing it to where on the X chromosome it was, and the possible location of it. And so we had done some work, but Dr. Bartlett, Greg Barsh's group and Chris Kalin's group has been working on this for many, many years. And they've actually known the mutation for many years, probably over 15, but they've been working really hard to try to figure out how the gene works,
Okay.
And so no one else has worked on it because we knew this was Greg and Chris's deal. So, there's not enough cat science to go around. So we share the love. And, but then Dr. Nakamura's group in Japan, they were developing a new reference for a cat from Japan. And so they worked on it as well. And so you'll see that really how this kind of worked out was Dr. Nakamura published his paper first in bioarchives, and then that cued Greg and Chris, we have to tell the world that we've been working on it too. And so you'll see that their paper came out the next day in bioarchives, but they were still trying to work out the function. And so they had to go to press with what they had, and Nakamura's group went to press with what they had. And now it recently came out in Current Biology, and both papers are in the same issue. And so then many of these news groups start picking things up. Like, so I've done an interview for San Francisco Chronicle and the Toronto Star just last week and stuff. And so they ask you, what is this mystifying thing? And that word actually came from Frank Nicholas, because Frank Nicholas is a bit of a historian for genetics. He also produces the online Mendelian Inheritance in Animals archive that lists all the DNA mutations for cats and dogs and everything other than humans and mice. Yeah. And so he knew this was something that was important since the beginning of genetics, and it really helps to present why everything you need to know about genetics you can learn from your cat. And
Yes.
so that's where this mystifying word, and everybody's picked that up since, but the color's been known for quite a long time.
I don't
Well,
think
that
Mendel
explains
used cats, though. He used little flowers.
peas.
He used peas, but
Right.
I was thinking about this, and this was terrible. I was like almost had a heart attack, and I was in the lab, and one of the students came in. So we have undergraduate students that work in our lab, and I was like, "Hey, how are you?" And she's, "Oh, I just took my genetics test." And I'm like, "Well, why? What are you moaning about?" And she's like, "It was about some guy that was like a monk and did peas. How boring is that?" And after I'm coming, Elizabeth, I almost have a heart attack. Then I thought, well, this is true. I have never seen a wrinkly pea plant, or I've never seen these pea plant. The 7 traits, I had to look them up, and I was like, I've never seen any of these. So how true? Why didn't you just use cats? So if you remember,
I think peas were easier.
peas were easier, and he was doing it because he was in a monastery. They were growing food for the monastery, and then also wine too, right?
Yeah.
And but his work was forgotten about for about 50 years. And then the
That's
turn of the
crazy.
century, in the 1900s, early 1900s, people rediscovered his work, and they're like,
Yeah.
oh, what else is there that has simple? So Mendel's traits were dominant, recessive, and co-dominant.
Okay.
And so everybody started looking around for things, and lo and behold, cats had things that you could clearly see that were dominant, recessive, and co-dominant. Recessive, like black, right? Dominant, like dominant white, and co-dominant, like white spotting. You have one copy, you're bicolor, 2 copies. It adds together, and you have the van. So here you had examples in cats. But then you had these cats that were orange, and it was clearly a bias towards males. More males were orange than females. And so that was the discovery that you could have genes on your X chromosome as well. And so that was somewhat the discovery of X-linked traits. And cats were right there at the beginning of it. So what's another key X-linked trait that people would have known about at the turn of the century? The Canadian might know.
Yeah, Canadian. Come
The
on.
Canadian should know because of her heritage with the royal family.
Oh, you're thinking of the hemophiliacs.
That's right. They were bleeders,
Hemophilia.
right? So hemophilia A is
Right.
also an X-linked trait. Yeah.
That
Yes.
Queen Victoria unknowingly disseminated through practically every royal
There you go.
family.
So we knew
in
there were
Europe.
kind of X-linked traits, but here's the cat, and the cat has something you can see. Boom, it's orange.
Yeah.
And so even though it wasn't a trait Mendel worked on, dominant, recessive, it is a single gene trait, and we tend to call single gene traits with simple inheritance Mendelian traits.
Mendelian.
And but cats added in the X-linked. Hamsters also have an X-linked orange, but it's a completely different gene in a completely different place on the X chromosome. So we've known for a long time that it's not the same gene.
Surprising what the article said. It is a mutation, the orange color. Is that correct?
Yeah, it's a mutation, as most all the colors are different mutations. So there's been a little bit of hype because there's like, why is it taking this so long to discover? Well, a couple reasons. So it's on the X chromosome. You would think, they'll find it right away. They'll find it right away. Well, the thing is, this mutation is not found in the coding part of the gene. And that's the easy ones to find, easier ones to find, right? And as we've developed genome sequencing and stuff, it becomes faster and easier and easier. So this mutation is not in the coding part of the gene. All right, that's a problem. So now you got to look in the introns and in the DNA between the genes. So now you're looking there. And then also, this is not a single like little base pair single change. This is like a 5 kb, 5,000 base pairs inserted, deleted out of this intron.
Is
And
that not
so
huge?
that's huge. Yeah, that's
Yeah.
gigunda. And so, but we have examples like this before. So white spotting in cats and dominant white, those are mutations in intron 1 of the KIT gene. And like white spotting is an 8,000 base pair insertion or deletion. So it's a little harder to find these big things that we call structural variants. And so they had been found. And as I mentioned, Dr. Barsh's group has known for quite a long time what the mutation is. And then Nakamura's group came along and they start a brand new project and they find it real quickly once they attempt to look and find it, right? So so it still leads us to, and then also there was no candidate gene. So obviously, we look for things. If you have something in white spotting, you look at KIT. You look at melanocyte ligand. So there's certain genes, PAX3. There's certain genes you know are associated with white spotting. But there was nothing on the X chromosome to say, hey, here I am. And so it ends up being in the middle of this gene called RGAP36. And we knew the gene did other things, but we didn't know it was part of the pigment pathway. So that's kind of novel as well. Now discovering that this gene is part of the pigment pathway, but every day we're learning that all our genes have multiple roles in different tissues in which they are expressed. So one gene is not just expressed in one tissue, it's expressed in different tissues, and it has what's called different transcripts. So it might have one more or less exon in a different tissue, and that gives it a different way to work. And so that was the fun of it all, is okay, now we definitely got it. And then Nakamura's group showed that it was a gene that underwent X inactivation, that was important because you see that in the tortoiseshell cat. And then Greg Barsh's group showed it was part of the pigment pathway. So together, the two papers tell a very
So
nice
back
story.
in time, does that mean that everything was black?
Yes, the mutation is new. So your wild type cat, an African wild cat, looks like a brown mackerel tabby, right?
Tabby.
And and so then this mutation came along and turned them into orange mackerel tabbies.
Right.
And so we think it's a mutation that's associated with domestication, because if you're, trying to keep cats around your house and you see an orange one, oh, that's cool, that's novel, that's new, that's my cat. And then everybody on the street knows, that orange cat belongs to, or ginger or yellow, whatever you want to call it, belongs to Bob
There
down
are
the street.
occasional orange females, though.
Well, sure, because it's on the X chromosome. It just means
Yeah.
that both X chromosomes have to have the orange
carry it.
mutation. So it's just a statistical game, is all.
Right. I was going to ask you earlier when you were talking about Mendel. Have you read the book called Cats Are Not Peas?
Yes, I have a copy. Guess who gave it to me? Mark Kittleson, as a going away present from leaving.
That's nice.
Yeah.
I'm not sure. I remember from when I was young and in early school that genetics was explained by the Mendelian trait. So Mendel was, of course, he's European. So maybe is that not the case in the US, that they teach them that?
Oh, absolutely. Okay. It's just this one student thought it was boring. Right. And I was like, well, it's historically, the thing is with the peas, you could you probably often get this. Well, I had a litter of kittens and no one was affected in that litter. So I must have a clean line of
Right.
kittens.
Oh,
And then somebody else says to you, every one of my kittens was affected.
Yeah.
Well, if you start adding up all those numbers, it'll end up with a recessive trait from 2 carriers that it was 25%. Well,
Mhm.
Mendel with his peas grew thousands of peas. And so he was able to define those segregation ratios.
Right.
And so those still happen in our cats as well, but you would have to grow a lot of cats to prove it.
Yeah.
Right. Right.
Peas are easier.
Yeah.
So it's not true that every genetic lab has peas growing, just to prove.
This is true. I've often thought at my house I should have peas, Arabidopsis, and I
Yes.
want a tank of glow in the dark fish. So,
Absolutely.
yeah.
So as a biology student, I learned with our friends, the fruit flies. We
Let's
should have
bring
done
up the
peas.
calico.
The calicos. Well, how do
Right.
the calicos work? So again, everything you need to know about genetics, you can learn from your cat. And in the 1950s to 1960s, a woman by the name of Mary Lyon, who is, I don't think she's a relation, but I'm going to try to track that down someday, So she was British, and she discovered X inactivation. And so women, we have 2 X chromosomes, but to compensate, so it's called dosage compensation. If you have 2 X chromosomes, well, you're going to produce twice as much protein as a male would, right? Well, so how we compensate for that is 1 X always gets turned off in every cell of our body.
Mm.
And that inactivation process occurs during early embryogenesis. And then once a particular lineage has 1 X turned off, that stays consistent from everything that comes from
And,
that lineage of cells.
and it's
And
random too, is it not?
and
It's
yes,
random.
it's random. Yeah,
Yeah.
absolutely random. And her early embryogenesis it occurs. And so that's why when you see a tortoiseshell cat, some of the hairs are black and some of the hairs are orange, because it depends which X got turned off. If the black orange got turned off, then the orange protein expresses, and then vice versa. Right. And then you'll notice that never changes. It stays a consistent pattern throughout the cat's life. Right?
Right.
And then
I think we should have kept both X's as women, I think, really.
kept both of them. Yeah. Well,
Yeah.
guys have their Y chromosomes, but we're still trying to figure out, is there any useful
What
genes?
the heck they do. Yeah.
Of course there are. You need the Y in order to start the differentiation of the Malarian ducks to become testes and
Eularian
stuff like that.
ducks.
Yeah.
Yeah.
So there you have a tortie, but then now you want a calico. Well, that's a
Yeah.
whole nother gene that gets
Yeah.
layered on top, and that's the KIT gene. And so those cats will have that 8,000 base pair mutation, big chunk of DNA put into the intron of intron 1 of KIT. And so what we think these big pieces of DNA do is they mess up either the splicing of the protein or the regulation of the protein, right?
Okay.
And so the KIT gene affects melanocyte production and migration. And
Right.
so KIT white spotting, all your melanocytes start at your notochord and then migrate dorsal to ventral. And that's why you have a perfect midline where you'll have a stripe where maybe one side of the cat's face is white or orange, and on the other
Yeah.
side it's a completely different color because there was
Yeah.
different X inactivation on either side of the cat, right?
They can be striking sometimes, right? When they're like,
Yeah, that's like,
Yeah.
so people think, oh, that's a chimera. No, it's not 2 cats put together. It's
Yeah,
just
yeah.
that's how our body develops. And so you could call this actually a midline closure defect, because it's actually the melanocytes are not migrating all the way to the closure of the midline. And that's little
True,
white
true.
spots and little belly spots. Those are basically like umbilical hernias and cleft palates. They're midline closure defects.
Right,
Yep.
right.
So,
Yeah.
so if this KIT gene also affects the number of melanocytes, that means there's not as many precursory melanocyte lineages. And so
Right.
one lineage has to cover a bigger area of the skin. So you'll have a
Right.
big patch of black
Yeah,
and a big
okay.
patch of orange. And so generally when you have calicos, you don't have brindled hairs,
Right.
you have
Patches.
big patches. And
Right,
that's
right.
because of the KIT gene. So that's called epistasis, when genes interact with
Epistasis.
one another, and so it shows you that a lot of our genes certainly interact and are part of complex pathways.
Yola might think of this too. When you say KIT gene, I also think of cancers,
Yes, that's right. Because KIT
right?
is it's a Karsten Ras. So it's an acronym, and it was one of the first genes associated as an oncogene, and
Yeah,
meaning a gene that we know is involved with cancer development.
Like
yeah.
mast cell tumors. So,
Mhm.
mast cell tumors.
Okay, melanomas probably, I would guess. You would think.
Well, cats with KIT mutations get more melanomas because they don't have pigments,
Right.
Right.
especially at the where you have the epithelial connections where the fur stops and the skin starts,
Yeah,
right? So that's
yeah.
where you'll have most of your melanocyte melanomas develop on the ears and stuff like that, but on those mucous membranes and yeah.
I don't know why it seems weird to me that one gene could do something as benign as make a cat orange, right? But also do something as evil, I guess, as, creating a cancer that might kill you. It just seems,
Yeah, absolutely. So we're finding that some mutations are just fun. A lot of mutations, most mutations are neutral.
Mm,
Some
right.
mutations you have kind of sitting around in your pocket and you pull out when you need them, like to fight off COVID or something like that, right? So you want
Yeah.
genetic diversity to be in there. But and then, we were also thinking that each gene made one protein.
Yeah,
That was
yeah,
the initial dogma. But now
yeah.
that's completely wrong. Each gene
Right.
makes at least three, generally about three or more transcripts, and those transcripts get turned into different proteins that act in different ways in different cell lines. So
Right.
when I was in school, we thought, oh, there's 100,000 genes. Well, no, there's more like 21,000 genes or so, but each gene makes many different transcripts and many different proteins. And humans have this RGAP36 gene. We
Yeah.
just don't have that weird mutation that turns us orange.
I wonder why not.
Because you have people with like red, orangey hair. That's not a mutation then.
That is a mutation, but that's in melanocortin receptor 1.
Ah.
And that's what makes yellow labs and red labs. And like just now, I rewatched the Preakness because I missed it. I was away, and the
Yeah.
one horse was Big Red horse. Big Red was Secretariat. That's a
Yes.
melanocortin receptor.
Yes.
So
Yes.
orange hair, red hair, Lucille Ball, Ron Howard, they have an MC1R mutation.
Different ways to almost get the same effect.
In fact, that's how we got involved with orange. Someone, actually it was Greg Acklin from Cornell, said to me one day, I want you to prove that MC1R has not been translocated onto the X chromosome, and that's the cause of orange for cats.
Okay.
And so that was our paper showing that melanocortin is not on the X, and
Right.
we didn't find the gene. We eliminated different areas. We said, so it's not here, so it could be there and there. And
Yeah.
that's the work we did for orange. Yeah.
Yeah, but you proved it wasn't the same orange as in
Melanocortin
red-haired
receptor.
people. Yeah.
Right.
Except from the hamster, any other animals that have this?
I don't think so. I think the hamster is it, and I think the gene is called SLY, SLY.
Right.
Yeah, but I don't know much about it. But yeah, and you have those kind of calico looking hamsters too.
You look at
Other
a gold-
animals have red hair though.
Yeah, and that's from melanocortin receptor.
Okay, so all the others are the same as red-haired people, you're saying?
That's right.
Like red foxes, that's melanocortin?
I don't know if that's been proven, but most likely,
Yeah.
yeah. Mhm.
Yeah, okay.
What about goldfish?
You got me on there. I'm not. I don't know my goldfish. Fish have all type of
I'm
extra
going to look it up.
chromosomes. They're tetraploidy.
Look
Oh,
at
they're
it.
they're
I'm going to look
complicated,
it up right now.
Fish are beyond
but
me.
but I bet there's a whole community of fish researchers. Like
Yeah.
if you think of koi and koi ponds, like it's a big
Yeah, exactly.
pastime.
Yeah.
Yeah.
I'm not sure how
And-
the pigment works either. The pigment
Yeah.
might be, it's slightly different in itself. So it might not be eumelanin and pheomelanin in lizards, birds,
Yeah.
fish.
Sorry, goldfish have melanophores and xanthophores, and the melanophores produce melanin, and for black and brown, and xanthophores produce xanthine and are responsible for orange, yellow, and red.
There you go. There you go. Now tell me you didn't use AI to come up with that.
As a matter of fact, I did. Sorry, we need to wrap up.
Ah,
As per usual, very entertaining and educational.
Okay.
yeah. I either forgot because I've hung out so much with you, Leslie. I've absorbed so much about genes, so I might have forgot that the gene for orange was known. So when these reports came out, I was thinking, why did it take so long to find like one gene? But it wasn't the gene; it's the mechanism that's been.
It's how it works. And still
Yeah.
we have some clues, but we're
Yeah.
not still, we don't know how it works at
Yeah.
this point
Work
in
to be
time.
done.
So they had to go to press. And remember, CC the clone cat
Yeah.
was the example of
Yes.
this that
Yes.
that showed that
Yes.
exon activation didn't get reprogrammed. And that's programmed by, if you recall, methylation patterns. Methylation changes and turns genes on and off, which is known as epigenetics.
Epigenetics, right? Well, I still have a list of things to talk about. So in 2 weeks, I guess we'll get to the
We'll be back. Yes, we'll be
we'll
back.
get
Okay.
to the rest of it.
I'm very excited, but for now, we say goodbye to the orange cat and Leslie.
This is the Purr Podcast, where we talk cats, laugh a lot, and learn something new every episode. Together, we bring you the latest updates, surprising cases, expert interviews, and just a touch of chaos, because nothing with cats ever goes as planned.
So whether you're a seasoned veterinary pro, a new grad, or you're just here for the cat puns, you've come to the right place.